Okay, welcome everyone. Maybe we give people maybe one more minute before we're gonna launch the webinar. We still have attendees joining. Okay, it's 12:01, so why don't we kick it off? So welcome everyone. My name is Boris Jürg. I'm a faculty at the Reagan and at Mass General. And I'm the associate director together with Doug Krakauer and John Lee, who's the director of the Center for AIDS Research Clinical Core. And we see John here as well. And so we had already one session of this two-session seminar series with the title Demystifying Clinical Research for the Early Career and Laboratory Investigator. People who might miss the part one was design and enrollment of clinical studies that happened in December with talks from Catherine Stevenson, Martha Jones, who joins us again today, and myself, just on some basics on clinical trials and how you establish those and how you set them up and run them. And this was all recorded and will be posted on the CIFAR webpage after we conclude today. And so today is the second part of the series, and we're going to talk about secondary use of collected samples. And we are very fortunate to have, again, three outstanding speakers today. John Lee will kick us off, followed by Athi Tsipras, and then Martha Jones again for part three. And I'm going to introduce each speaker right before their talk. And so let's just start with John, who is an associate professor of medicine at the Brigham and at HMS. He actually runs an active clinical and translational lab, and his research is really focused on viral persistence, reservoir, and resistance. He holds several director positions for virology, a specialty lab at the Brigham for the ACTG Virology Core Labs, and is also the vice chair of the ACTG Cure Transformative Science Group. And so John is going to talk about identifying HIV clinical collaborators and samples. Please.
Thank you so much, Boris, and thank you to everyone who's able to join today. Let me just make sure this is working. There we go. All right, so I just want to first of all mention that this QR code on the bottom right will take you to the CIFAR Clinical Core landing page, where you can find a lot of the the actual kind of slides that we'll be showing, but also some of what we're talking about in this talk today. So some of you here today might be outside of the HIV field, and if you are, I think we're thrilled that you're here. While my particular talk today is focused on finding HIV collaborators and HIV samples, I just want to mention that our next couple of talks by Athi and by Martha are going to be focused on practical issues in working with a collaborator and also IRB issues for secondary use. And so those have broad applicability even outside of for HIV field, and so for laboratory and early career investigators. So stay tuned, even if you're not in the HIV field. So I want to first of all mention that the CIFAR is, of course, made up of institutions from across Boston. And to introduce the CIFAR Clinical Core leadership, which includes Boris, who you just met, he's at the Reagan and MGH, Doug Krakauer at the Fenway and BI, myself at the Brigham, and then Sandy Burchett, who's at Boston Children's Hospital. The Clinical Core helps facilitate and support CIFAR investigators with a focus on junior investigators, really trying to help them get their studies off the ground. This may involve helping them with some research assistant support, regulatory help, or just connecting them to the right collaborators and biospecimens which is what I'll be focusing on today. We also help put on seminars like the one today and help provide study and or career mentorship and advice for junior investigators. So if you all have questions about finding a clinical resource, a collaborator, or any other questions, don't hesitate to reach out to Boris, Doug, Sandy, or myself for help. So that's what we're here for. So for today's talk, finding clinical collaborators and samples, there's really two big main sections. One, I'll be talking about kind of local collaborations. So really going through what we have on the CFAR website and giving you an example from the HIV tissue study at the Reagan Institute. And then I'll go through some of the national biorepositories that are, I think, underutilized and have been really important both for my own career and I think have a lot of potential for all of you out there who are looking for samples and collaborations. So if you go to the Clinical Core website, you'll see a couple different links, clinical trials and also cohorts. I'll start off by going over to the cohorts site. If you click on the cohorts, what you see is a list of clinical cohorts that are led by Harvard CFAR kind of PIs. There's currently 25 of them on the list, and we try to keep them up to date by contacting PIs on a regular basis and making sure that the list is up to date. And I want to show you what kind of information that you can get from this site. If you click on, for example, this HIV tissues website link, what you get is information about the PI here, in this case, Doug Kwan. You'll get the contact information, study participants, enrollment details. We also give the PIs and the study coordinators an option to record a video. And I'm actually going to play this video here. This is a video from Daniel Worrell, who leads clinical research at the Reagan Institute. And just make sure this works here.
There's no sound for us, John. No?
Oh, shoot. There is no sound. All right. Well, there is sound on my computer, but I guess there's no sound where you are. But pretty much he is, Daniel is explaining, Daniel is explaining, explaining his, the study, the participants who are enrolled, the types of samples that are available, as well as how to get in contact with folks. So, go onto the website, click around, find some of these videos and some of these studies and really I think there's an opportunity for you to see what is happening around the CFAR PIs and at the different institutions. All right, so I'm going to keep on moving here and go to the next slide. So this is just a list of the publications that have actually been have actually come out of this HIV tissues repository. And I will say also that our next speaker, Athy Sebris, actually runs one of these cohorts, the HEAL study of ART suppressed individuals. And I'll let him talk about a study, but that's another great example of a cohort study with samples that are available for collaboration. All right, so if you click on clinical trials, what you get is a link to ongoing interventional clinical trials. Now, I will say that it's a little bit more challenging to aggregate all of the interventional studies and trials in one place. And so what you see here is actually a link to the to some of the hospitals, the websites, landing pages of the hospitals across Boston. In addition, if you click on the clinicaltrials.gov link, you'll find that there is actually a link here to a list of all of the ongoing or planned interventional HIV trials occurring in Harvard CFAR-affiliated institutions. And when I did this recently, what we get is that it sends you over to the clinicaltrials.gov site, and you see that there are 24 studies that involve HIV that are either planned, so not yet recruiting, or currently recruiting, and are being performed at the Brigham, MGH, Beth Israel, Fenway, Children's Hospital, Reagan, or the Dana-Farber. And if you click on the first one here, what you see is that this is a comparative effectiveness of individuals versus group level interventions to reduce HIV and STI incidence. It tells you the PIs, and there's a lot of information here about the study and its start date, completion date, and its where the study is at the moment. So again, the clinical, you can find the links both to the clinical cohorts as well as the interventional studies on the clinical core main website on the CFAR website. All right, so I'm going to move on to the international trial. So I'm going to highlight two of them, the ACTG as well as CNIX. So the ACTG stands for Advancing Clinical Therapeutics Globally for HIV/AIDS and Other Infections. Used to be known as the AIDS Clinical Trials Group before they rebranded themselves. There are several areas of focus for the ACTG, including HIV comorbidities, cure, emerging infections, treatment, TB, and hepatitis. And if you look at the overall kind of organization of the ACTG, there's Leadership Operations Center, there is a Lab Center, there is a Statistical Data Analysis Center, and there are a number of different transformative science groups based kind of focused on the different areas of interest for the ACTG, including ART, reservoirs, remission and cure, hepatitis, comorbidities, TB. There's also a Women's Health Collaborative. One of the reasons I really wanted to highlight the ACTG is that so many of our CFAR colleagues are actually in leadership positions within the ACTG. And I just want to highlight, for example, Raj Gandhi at the MGH, who is actually the vice chair for the entire organization, and he'll eventually take over as the chair. I am the director of the Virology Core Labs, as well as the vice chair for the Reservoir Remissions and Cure TSG. Serena Koenig is the vice chair for the ARTS TSG. You got Arthur Kim, who is the chair of the Hepatitis TSG. Markella Zani is the vice chair of the Women's Health Collaborative. And then Michael Hughes heads the Statistical and Data Management Center, and he's at the Harvard School of Public Health. So there's, really, I think, whatever your interest is, you can come to one of us for help in terms of connecting you with the right people and the right studies within the ACTG. So also within the ACTG, I just want to show you within the Cure TSG some of the planned and ongoing ACTG Cure studies. And you can see there's a number here that involve latency interruption or latency reversal, immune therapy, including bNAbs and therapeutic vaccines, and also there's AAV delivered bNAbs and CAR T cells. Now, I will say that these studies are really ongoing or kind of recently completed. And so samples may not yet be fully available for these studies, but there are a lot of studies where there are available samples. And these include, this study from 5321, which is an ACTG HIV reservoir longitudinal follow-up cohort of ART suppressed folks that Raj actually leads. I've got a treatment interruption study, 5345, that I lead. And then there's studies of early treated individuals. There's study of HIV controllers who received ART. There's studies on latency reversing agents and immune modulators. There's studies of isotretinoin for immune. This is just an example of some of the studies as well as some of the PIs who are leading this study. So a lot of samples that are available that the ACTG wants to give you. You just have to fill out what's known as a new work concept sheet or a data analysis concept sheet. And pretty much all of the samples get sent to a repository, the ones that are not analyzed immediately, at least. And so if you've got an interest, you just have to fill out one of these new works concept sheets or a data analysis concept sheet in order to get access to the samples and the data. And if you look at the ACTG biorepository, there is an online website. Again, these slides will be available online after the seminar. There are now 3.8 million samples from 29,000 participants. So a lot of different samples that are available for folks. And then again, my last couple slides, one here is that there's the CFAR has a CNIX longitudinal kind of cohort that is based off of 10 different sites, including the Fenway, and 48,000 patients enrolled, really diverse group of individuals and participants, and linked to specimens and available to investigators. And you can contact Ken Mayer at the Fenway for questions. So I'm going to stop there and throw it back to Boris.
Thank you very much, John. That was great. Just looking if there's any comments or questions at the moment. So please put them in the chat, and then maybe John can answer some of them already. Otherwise, we're going to have some time, hopefully, at the end to discuss questions once they come up. Okay, so let's move on to our next speaker, which is Athi Tsipras, who's also an associate professor of medicine at the Brigham and HMS. And Athi is also interested in HIV reservoir, HIV-1, but also HIV-2 persistence. He actually leads, as John already mentioned, the HEAL cohort, and I think he's going to talk about that a little bit more. But also chairs interventional clinical trials for the ACTG and is also the director of the development core for the Harvard CFARM. So Athi, take it away.
And hold on one second here. Great. So thank you, Boris. Thank you, John. It is a pleasure to join all of you on this chilly afternoon, at least here in Boston. For those of you who joined the clinical course seminar last week, the focus was on clinical trials. You may have found that slightly intimidating. Clinical trials have many moving parts. They involve many moving investigators. But the focus for today's session is on establishing collaborations for the secondary use of samples that have already been collected. This should be in many ways a more straightforward process, and I hope as we give you some practical advice, something that everybody who is listening will feel comfortable at least attempting on their own. So the goal of my talk here is really to talk about how do we establish these collaborations and how do we nurture these fuzzy friends here that we're referring to as birds and make them blossom into some really fantastic science.
Athi, can I just interrupt you for a second? You're still in presenter view.
Try again. Is this any better?
Yeah, this is good. Perfect. Thanks.
Fantastic. Apologies. Okay, so just a brief roadmap here. We will cover the different types of collaborations. Again, today's focus is on secondary use of samples. We'll use a vignette to talk through approaches that work well and perhaps may work less well. We'll talk about special circumstances that can arise as you start to try and establish a collaboration. I will emphasize that there are bureaucratic and regulatory rules that we must follow. They are different from country to country. We'll focus on those here in the United States, and then emphasize at the end the value of thoughtful communication. So as I mentioned, there are many different types of collaborations. Perhaps you need data, perhaps you need expertise, perhaps there is intellectual property. Someone has a technology that you need. These are all very appropriate types of collaborations. I emphasize here at the bottom, community collaborations. Ultimately, you do want to understand how your research is being perceived and how people may be willing to engage in your research programs. None of those will be the focus of today's talk. What we'll focus on today are how we establish collaborations where samples are shared between investigators. So who typically are the people who are involved in this process? Okay, so let's start here on the left, where at the top, we have a group of principal investigators who have established either a cohort or run a clinical trial. They have done this here with the schematic in the middle, where they have a team of researchers or researchers' assistants, and they have interacted in a research study format with participants who have donated these samples. And the goal for today is we're going to try to figure out how do these people communicate with our protagonists who are the prospective collaborators. Any resemblance between any of these two characters with myself is purely coincidental, but we want to figure out how can we get these two groups to communicate effectively and then conduct high quality science. Keep in mind here that a good collaboration is going to benefit everybody. Nobody wants to be hoarding their chocolate. For the most part, and the IRB will make you designate this in consent forms, the majority of the studies that are conducted, at least in the HIV reservoir or latency or cure space, are really given out of altruism. There is no direct benefit expected to the participants. And so at least to me, this creates an obligation to maximize the value of the samples. If you're not sharing samples, you're having difficulties establishing collaborations, it is important to ask yourself why. Most of these cohorts are set up, yes, so the investigators can pursue their science, but more generally, so we can advance the science for the betterment of everyone's health and the public health. I will make one plug for my particular cohort study, and we'll use that in the vignette, just to point out that here in Boston, we have established this cohort of about 100, 120 participants, most of whom are treated and suppressed. These may be participants with HIV-1, HIV-2. There are longitudinal blood samples. These may be peripheral blood samples. They may be leukapheresis samples. We have up to 10 years of follow-up. And if at any point somebody is thinking about, hey, these samples may be of use in my research, please don't hesitate to reach out. Okay, so let's run through what may be the optimal approaches, or as we'll see, not the best approaches in a vignette here. And we'll start off, somebody approaches me, Dr. Blodwin, and they've heard about my cohort. The biorepository sounds like an amazing resource, and I'd love to talk to you about a potential collaboration. Okay, so this sounds like a very nice way to begin, and our mood here is generally quite positive. We like to hear more from Dr. Blodwin. Dr. Blodwin has an interest in how CD8 positive T cells and NK cells contribute to reservoir control. But Dr. Blodwin tells me, before I know what samples I want, could you create for me an Excel spreadsheet and summarize the following data parameters, and hands over a list of about 20 or 30 variables. Okay, so now I'm not feeling quite so shiny about this arrangement. You were creating work for me. You haven't really explained to me what your idea is. I'm not really sure how I feel about this. Dr. Blodwin, however, continues. If you don't already have HLA genotypes on these participants, could you get them? That'd be great, thanks. And this probably, I would guess, is somewhere between a $10,000 and $30,000 cost to me. And so at this point, we have some bad blood. So what has gone wrong here? Fundamentally, one of the researchers is conveying their needs, but they are talking at the potential collaborator rather than with the collaborator. So how do we avoid this? These are some really very basic points here. Really explain your research along with what the knowledge gap is. So in some ways, as you establish a collaboration, you're thinking about things in terms of NIH grant or in proposal language. What is the knowledge gap here? That helps orient the collaborator as to what really here is important. How do their samples help? Sometimes it's useful to emphasize the novelty or the high priority nature of what you're doing. And if your ideas are unique, that can be helpful. Sometimes if you're really just starting out your research program or your lab, if you can drop a big name somewhere, oh, I talked to Dan Baruch about this, he was very interested. Oh, Bruce Walker said this was the most important thing he heard of in years. That can actually help bring people together. Most collaborators will collaborate you regardless of this seniority or power structure. We'll talk more about that in a second. But anything you can do to start this collaboration is worth trying. At the end of the day, we are all people. So building rapport and listening is very helpful. Is there something that the sample owner, the person who runs the cohort, do they want something? Do they need something? And really, if you're at a loss for exactly what to say, you are obviously excited about this research. See if you can pass that on to the people that you are speaking with. Now, the question is who exactly you're going to collaborate with. As John said, there are these federally funded national groups here in the US. They have millions of samples, in some cases that have been sitting in freezers for decades. So the pros of working with these national groups are, as we pointed out, many samples. You may find that a lot of metadata has been generated by other investigators who have used these samples. Perhaps that will be useful to you. And these cohorts, as I mentioned, are very likely to want to collaborate. The downside is you're going to follow their process. And as John pointed out, there may be things called new work concept sheets. There may be reviews. These can take some time, 3 months, 6 months, 12 months, all would be very expected. On the other hand, you may think about using these principal investigator-centric cohorts. They can offer local access, or as I put it here, boutique access. The advantages are you can forge stronger individual scientific connections, and the processing and the passing of samples may be more efficient. The downside is these investigators may have fewer samples, or for one reason or another, they may just be less willing to collaborate with you. Do collaborators and collaborations, do they cost anything? So generally, no, but there's a few things to keep in mind here. It has taken effort to collect these samples, so the sample provider may make a request for authorship or acknowledgement. And if this is part of a grant proposal, they may express an interest in some modest amount of effort for them or somebody on their team to offset the cost of the sample procurement. If you go to try and establish a collaboration and someone says, that sounds very interesting, that'll be $20,000, please. That would be a little bit more unusual. And in those cases, perhaps you can approach a more senior investigator to see if they have advice or can maybe explain why is it that this person is asking me for money up front. What are those special circumstances that I referenced here at the beginning? So many groups within Harvard and outside of Harvard are interested in collaborations internationally. Very important component to the HIV research agenda. Just a note here, beware of scientific colonialism. What does this mean? Many of you will be familiar with this. Colonialism, generally refers to the concept of people coming to a country, extracting some resource from that country, and offering nothing to that country in return. Historically, this is bad. It is no less bad now. And so what you may find out approaching collaborators is that all the scientific work may actually need to be done in country. Perhaps they may require you to contribute something to the training of scientists in these countries. It may be actually practically impossible to get samples out of those countries into your lab, and that is an entirely reasonable approach, something you should expect. If you are able to establish one of these collaborations, you should, in the United States, prepare for federal import requirements. I established an HIV-2 collaboration in Mali, and prior to any samples arriving in the United States or in my lab, the CDC has to review my permit process and make sure that everything is being done in accordance with federal regulations. Much as perhaps in our personal lives over the years, this is also true in science. Some people may just not be that into you. And fundamentally, this boils down to your mission is not their mission. Some international groups, their programs will have clinical focuses. When their resources are limited, research really may not be their top priority, and this may be for many completely legitimate practical reasons. For rare samples, the barrier to collaboration or the price of collaboration may increase. It may be that these samples were acquired after a great investment of time and resources in an area that nobody else has been able to establish a collaboration. In these cases, the requirement may not be authorship, but it may be specifically senior authorship. And you should expect that the more invasive the process to obtain the samples, somebody is taking biopsies of tissues, lymph node, gut, the greater the price. You may encounter situations in which, for these rare samples, you just cannot get access to them. They're already spoken for. These samples are available, but your idea is not a high priority enough. Perhaps they're sending these samples to their friends. That happens less commonly, but having common points of reference can sometimes be helpful. In general, the more rare a sample gets, you do want to emphasize the novelty of the science. So you're not going to pitch just a descriptive idea unless you absolutely have to. And if you do, then you want to make this more novel in some way. In this case, I've listed here, perhaps you pair this with technical innovation as a way to pique the interest of the cohort's PIs. We do want to just mention how to collaborate across differences. And I'll just make the point here that what you mean or what you say is not always the same thing as what someone hears. So we do want to carefully consider how these differences may affect the dynamic in terms of seniority of investigators. Perhaps there's a power differential. How does gender factor in? What role may race be playing as you're attempting to establish a collaboration? Overall, one thing here I just want to say on the regulatory aspect, Martha Jones will speak about this in a moment more authoritatively, but the general rule here is follow the rules. You should think about whether you need a material transfer agreement if you are sending samples outside of your institution. And really, you do want to submit for an IRB determination every time you're thinking about doing research like this. If you do not make time for the IRB, they will make time for you. They will give you time to sit around thinking after they shut down your research program for not doing things correctly and ask you to take some corrective action. That is not the main function of the IRB. That happens very rarely, but follow the rules. Before you start using samples, make sure you have an IRB determination. Perhaps this is not human subject research. Great. They are the ones who are going to tell you that. And then just make sure that how you plan to use the samples is consistent with the original consent process that generated the samples. And here I just draw your attention to genomic analyses, make sure that that was specifically mentioned. For those of us here in MGB, you do want to make sure that you update your lab's institutional biosafety committee filing. If you're an HIV researcher, now you want to work on measles, make sure that's added in there so they know what type of research is happening in your group. So this here is my second to last slide. It's really a summary slide, just to really go back to the basics here. Practically speaking, good communication leads to good collaboration. So just be mindful of the effort it takes to collect samples, something that was missing in our vignette. And really, nobody wants to feel like they're your butler. So if I show you the picture here, if you're acting like a bull in a china shop, take a step back, refocus, and get back to a little bit more quality communications. You want to use the samples as you said you would. You want to be transparent. Perhaps there's delays, perhaps there are problems. Advance the science, enjoy the collaboration. And just to point out here again, the goal here is to have the principal investigators of the cohort and the collaborators working well together, and everybody wins. So that's what I have for you here today. I just do want to make one last note on a slide here. I am joining you on this call. I also participate in the Harvard CFAR. I help co-direct the developmental core with Molly Franke. What does the developmental core do? We are the funding arm of the Harvard CFAR. We fund early stage investigators locally and internationally, but we also offer mentoring opportunities. This may be mock grant reviews. These may be research in progress meetings. And we really are stronger when the entire diversity of the Harvard CFAR and our international collaborators works together with us. So if there are any questions about funding opportunities, please do reach out. I've appreciated the fact that we had a chance to talk today, and I will turn things back over to Boris. Thanks very much.
Great, thank you so much. That was awesome. Let me see, there's questions, but maybe we can keep them for the panel discussion at the end. So then let's move on to our next and last speaker. We're very glad to have Martha join us again. Martha Jones is the Vice President of Human Research Affairs at NGB, and has a ton of experience and national recognition in human research protection. I could go on and on and on of all the achievements, but maybe we just jump right into the topic, if that's okay with you, Martha.
Yeah, absolutely. Thanks, Boris. Let me see if I can get back here a little bit. There we go. Thanks so much for having me. As was said, I'm going to dive a little bit deeper into the IRB considerations for the use of secondary samples. But as you might have determined from the comments, this is a huge area and often very nuanced, and we look at things case by case. So I'm going to give you a very high overview of this, but certainly happy to talk with anyone that has specific questions. So just as background here, there are actually a lot of factors that we consider in setting up how we approve or determine that samples can be used in secondary use research. There's a large set of historic events that we consider, as well as current public perception about how research should be happening. It's very important for us to continue to grow public trust in science, and part of that is in how samples and data are used in research. There are certainly federal regulations and specific MGB policy that dictates how materials can be used in research. Part of this comes from the federal regulations around what is a human subject. As you heard, the status of the consent from the patient or research participant is key in determining secondary use. And we also have an MGB policy on specimen and data transfers and access, which we assess on a case-by-case basis as needed. That policy is enforced through a committee called the Data Tissue and Sharing Oversight Committee, and there is a process for approval through the Chief Academic Office for certain cases for secondary use of specimens. We also look at the scientific merit of the proposed use and the context of the engagement. So the collaborative nature and the requesting party is an academic or a for-profit entity that we're collaborating with, financial considerations, and then the IP issues, products, licensure, those kinds of things that are all considered. So with that background, I wanted to give you a a basic of where we started in thinking about consent for secondary use of products. Often there, and I'll show you in a minute, there are several ways that samples become available. But we've taken as kind of our basic what the federal government developed under its genomic data sharing policy, which set a clear date of before and after approaches to use of materials. That date was 2015. We don't necessarily use that particular date, but in general, we think about, was there a time before in the past when it maybe was not standard for us to get consent or get specific consent for future use of materials, whether it's data, specimens, images, etc. In the GDS policy, it notes that as an institution, we can make a local decision about whether use or contribution of those materials is appropriate. Going forward, though, the expectation after this date was really that collection of materials would be done with consent, specifically in the context of using them for whole genome or whole exome sequencing. But we tend to apply this more generally across the board with regards to any use of materials that maybe perhaps did not have consent. So that's kind of an underlying federal policy that we use as a basis for some of our decisions here. But you also need to remember, first and foremost, samples that are collected here, created here, are owned by the institution. They're owned by Mass General Brigham, not by the individual investigator that may have collected or curated or derived them, unless that ownership is otherwise assigned through an agreement process. So, for example, in a clinical trial that's sponsored by a pharma company, typically the language in those agreements will say, even though we are collecting samples, they are actually owned by the pharma sponsor because they are paying for the collection of those. So you need to understand who actually owns the materials that you propose to be working with. As was stated, samples coming into or going outside of your institution must have an agreement associated with them that's executed through one of our MGB agreement offices. This can be an MTA, it could be other types of agreements. That agreement language could be in a subaward for a grant, it could be in a sponsored research agreement, a variety of different types of agreements that might apply to that exchange. And then often when we're working with researchers, because we work so closely in a collaborative way with many of our Boston affiliated institutions, we sometimes forget that they're actually external institutions. So just remember that entities external to MGB, we still need agreements with, even though we work closely with them. Examples would be Harvard, Broad, Dana-Farber, external collaborators. And we also have a situation where we have researchers that have dual appointments. For example, they have appointments both at MGH and at the Broad. And if they are basically sharing samples with themselves as they put on their Broad hat, there should be an agreement in place, even though they have that dual appointment. So let's jump into really the IRB oversight. And I mentioned the idea of whether something is human subjects or not. In 2018, and it really became effective in 2019, there were new human subjects regulations that went into place. And both the federal HHS regulations as well as the Food and Drug Administration specifically identify that when you are working with biospecimens, that this is considered human subjects research. The exception is if it's HHS regulations that apply to the study, that allows for you to de-identify the biospecimens, remove all the explicit identifiers. And in those uses, it may be that the IRB can determine that you're not actually conducting human subjects research. The kicker here is that if the FDA regulations apply to your research, it may not matter whether you have identifiers attached or not. The FDA doesn't put the emphasis on whether there are identifiers attached or not. So it's very possible that you could be working with completely de-identified specimens, biospecimens, and yet the IRB would still consider it to be human subjects research. So as you heard earlier, it's best if you try not to guess yourself about whether you're conducting human subjects research or not. The IRB has an application process where you can get a formal determination, and they will make considerations about what regulations apply to your research, so that you don't go down the wrong path there. So here are all of the types of or situations of consent that we run into, depending on the source and the different types of consent. We treat each of these areas a little bit differently when we're deciding as an IRB whether to approve secondary use or not. So starting with a common source is discarded or excess clinical specimens, or it could be derived data or clinical data that a researcher wants to use. Of course, in those situations, there is no research-informed consent that the patient provided for use of their specimens in research. Sometimes the IRB will determine that those uses are not human subjects research, or they might determine that they are research, but they fall into a category called exempt. We also have data and specimens that are collected specifically for research purposes, and you've heard about a lot of those. This can come from disease specimens, normal, or it may be that specimens extras are collected during a clinical procedure specifically for research purposes. Similarly, could have a situation where there is no consent or it's been waived. A lot of times we have very old research where we don't have a record of what the consent was. Unfortunately, it's been sitting in someone's freezer for many years and it's not connected to the consent. All of those gray areas here are considered unconsented samples. A better situation is where we have consent, but perhaps the consent was not specifically for the current or secondary use that's being requested. It was consented for another purpose. Sometimes in that situation, the consent is silent. It doesn't say one way or another whether it can be used in the future. And in other cases, the consent actually has prohibits use for other purposes in some way. So we look at that when we're reviewing consents. Best of all worlds, for secondary use, when you look backwards, you have a consent that clearly identifies it can be used for future research and or for the specific secondary use that you would like to use it for. Again, these new federal regulations that I talked about that became effective in 2019 have put us on a path to have a better situation for future use. They actually require notification of future research use. So I think our consents today are better than they were even a few years ago. They have to specifically state about biospecimens, whether they can be used for future research or distributed to other researchers. And they have to talk about with respect to whether identifiers would be included or not. Whether or not it can be used for commercial profit and whether the subject will or will not share in that commercial profit, and whether there will be whole genome or exome sequencing involved. So last slide here, I just wanted to go over some key considerations when you're thinking about secondary use that you might want to walk through before you actually apply to the IRB. First of all, what is the original source or sources of the samples that you want to use? A lot of times, especially if they're coming from external sources, our researchers don't actually know the original source of the samples, and that's key for us to know in the IRB. Were the samples collected with consent? If they were, what we are looking for is not that there was just a signed consent document, but was there specific language that describes the potential for future use or the potential for secondary use beyond the original collection purpose? Was there any language that prohibits the proposed use? So sometimes we see things like it will be restricted to only being used in the future for certain diseases, Or sometimes it says that it will only stay in the lab of the PI and no one else will have access to it. Unfortunately, there is some very restrictive language that we see in older consents, and we have to pay attention to that as well. If there was not consent, then it's really critical that you provide sufficient justification, scientific justification for conducting the secondary research without consent for the IRB to consider. It's also important to know the dates of collection of the samples that you would like to use. Often what happens, particularly in repositories or studies that have been going on for a long time, the consent document will change over time. What we'll see is that original consents did not have any language about sharing, or they might have had restrictive language, but the consent changed over time. And as newer samples were collected, there is appropriate language. So the IRB might be able to approve your use of samples collected, say, since 2020, but samples before that may not be usable for secondary use. The IRB is not going to basically override that agreement that's made with the participant in the consent process. So they will not give you a waiver of consent if they see something in the consent that would be inconsistent with what you propose to do or that would prohibit that proposed use, even if the person is deceased or you would remove identifiers. Those terms of the consent live with those samples continuously. And then in most cases, you will need to submit what we would call a secondary use protocol for the specific project you are proposing to conduct. You would need to cite the original source of the samples, as I said, including the IRB number if it was an MGB protocol. Again, don't try to use secondary samples under a repository protocol or other umbrella protocol. That's no longer being approved by the IRB. If the samples are coming externally, details on the source, the consents, and the terms of use, if any. If internal, the IRB will look at the protocol under which they were collected and review the consents to see if your proposed use is consistent. And then again, if there's no consent, the IRB will consider a waiver of consent, but there may be limitations, particularly if sequencing is proposed in your protocol and if you propose to collaborate externally. So I will end there and turn it back over for questions.
Fantastic. So that concludes the 3 talks for today. And it leaves us 10 minutes for a little bit of discussion and answering questions. And there are quite a few actually occurred here in the discussion window. I think one, well, let's just go through it. One was from Malika to John about how one can access actually either samples, specimens, and or data, I guess, either from what is available here within Boston, which is a little bit more difficult, but more from the national networks or international networks like the ACTG. So John, do you want to comment on that a little bit?
Yeah, so I think, and I think specifically she had a question about participants who have substance use disorder. And I think locally there are a number of investigators who are working with those types of participants, both at BU, but also here. I think actually Athy may be working with one such group of participants, so he can comment. In terms of, and again, the local ones, you would just have to contact the PIs and kind of work things out as Athy described in his talk. For the ACTG, you know, I put in the link to the biorepository on the in the chat. I wish the ACTG website was a little bit easier to navigate, but it is not. And actually, Ron had a comment here as well that not all studies are even part of the biorepository website. The ones that are still ongoing, longitudinal follow-up studies that have really interesting samples and data and samples that they want people to use. Well, actually, some of those are not even on the website because they're still ongoing. And I think the best way to navigate the ACTG and CNICS is probably to find a Sherpa like myself or Raj or Ron or, you know, or someone who's affiliated with ACTG or Ken or Doug Krakauer for CNICS. and just ask them to show you the ropes, because it involves kind of knowing, the study population that you're interested in, which studies for the ACTG enrolled those participants, which and then how do you navigate the NEWIRS concept sheet and data analysis concept sheet kind of pathway, which is actually not that difficult. It's actually fairly straightforward once you know the process, once you know which samples you're looking for. And so I would say just contact one of us and we can help guide you through it. Athy, did you want to say anything about substance use disorder and potential cohorts that are available?
Yeah, I would say so. We do have a cohort of injection opioid users locally here in Boston. One of the things you're going to find with these cohorts is that there are practical limitations, and the main limitation is the volume of blood that can be consistently drawn or provided by people with HIV and substance use disorders. So this limits the amount of samples that can be placed into a biorepository. I'm happy to talk to Malika or anybody else. I can tell you, if you're interested in PBMCs, almost all of them have been used up. My experience is not necessarily what you may find in other cohorts, but if you're interested in plasma or serum, more likely to be available through my cohort or anybody else's cohorts. So it is. There are other challenges which Malika, we can kind of discuss, but it is an important group that people need to study more.
Yeah, just one last comment there. The ACTG, for example, has a hepatitis, TSG, as well as studies of individuals who are kind of non-adherent to their regimens. Both of those groups, those folks who are the hepatitis C studies, and then the individuals who are not fully adherent to the regimens, I think may include individuals who have substance use disorders. And so I think there are these kind of studies where you could find very specific participant populations. But again, I think finding someone who can help you navigate that process would probably be best.
And just a comment on the requesting us samples or data or specimens from the ACTG. I mean, it's not the fastest process overall, because these concept sheets have to be reviewed and are being reviewed. But at the end, it's really worth the effort, because it's a really unique resource of getting like excellent specimens in data with enough metadata attached to it that I think doesn't really exist much outside of this network. Definitely worth, first of all, looking into what studies have been done and what is available, and then really utilizing that. And there's a strong emphasis from the ACTG actually that all these specimens that they're banking for many years are pretty much used for research. So one question came for Athy regarding when one should actually discuss authorship with a clinical collaborator. When is a good time? You don't want to obviously start with that, but it's also not a bad idea to bring that up before all the work has been done, and then there's kind of some fight afterwards. So what would you recommend?
Yeah, Boris, I agree with you completely. Not the first line out of your mouth, but I like to transparently discuss that early on. It is interesting that no matter how far along you are in your research career, authorship ranking orders generates the most amount of distress. For secondary use of samples, this should be relatively straightforward. What I will tell people is, hey, your idea sounds great. Write me up a short paragraph, and please just include me as a middle author somewhere. Sometimes I break the ice with a joke, saying, I demand to be eighth author or something. That tends to lighten the mood. I would talk about this certainly before the work is done. Again, if your samples are very rare, one of a kind, or there's only a few left, perhaps you should have an expectation that the seniority of placement of the author may change. But I would talk about this really after you've cemented an idea. Say, hey, this is good to work. This will be good to work together. Just fire off a quick email. Okay, just to make sure we talked about this, and you're going to place me as a middle author and tell me where to send the samples. I think that works really well and avoids problems down the line.
There's one question from Kimmy Park. When submitting initial review, how can we navigate scenarios where secondary use study staff overlap with the source study staff regarding de-identification of samples and associated clinical data collected as part of the source study protocol. I guess that's for Martha.
I think that's me. Yeah, this comes up. So the best way to set this up is if you have a source protocol, is that you limit the number of people that are on the protocol. For example, if that's a repository protocol, that protocol should only be the people involved in the collection and storage and distribution of the samples. Anyone that wants to use those samples would then submit a secondary use protocol. Now, if someone that is on the research team of the source protocol also wants to use those, they are by nature considered to have access to identifiable data by virtue of being a research team member on the source protocol. So any use by those individuals would be considered use of the materials with identifiers, regardless of whether they actually need identifiers for their use or not. They would still be submitting a secondary use protocol for their particular project, but the IRB would see them as having access. There's not really a way around that, and that's based on guidance from our federal regulators, so we don't have a lot of flexibility there. But again, ideally limit your source protocol to the minimal number of people that need to do the collection and management, and then all others that want to use it can, for example, if the users are not on the source protocol and they don't need to have identifiers, then their use may be determined to be not human subjects research.
Great, thanks so much. John, you had a question for Ron?
I think Ron had, I think Ron largely answered the question in his long message that we pasted onto the.
I didn't know if IDU can be searched for.
Oh, that one. Yeah. So the biorepository website has a limited. This is kind of goes back to the fact that a lot of information is not readily available on the biorepository website. You can search for things like demographics, HIV status, CD4 count, viral load. There's a lot of things, but there's obviously a million different kind of covariates that people are interested in, including IDU. I actually, after Malika sent that message, I went on the biorepository website and couldn't find an IDU or substance use on that as a search term. And so I was wondering whether Ron knew whether that could be searched for. And because they've got, so Ron, for those of you who don't know, is part of the SDAC, or the Statistical Data Analysis Center for the ACTG. So they do a lot of searching of samples based on various criteria, and Ron has replied that they do collect IDU data. So again, so that is a good resource. Thank you, Ron.
And I don't see any other questions, but I think overall, hopefully this was helpful for people to get a little bit of insight. There's really not much magic here. This is all totally doable, and I think Boston is really like a fantastic resource in terms of people, cohorts, and research to do whatever you want to do. You know, all the contacts are you can find us easily on the CIFAR webpage, and just please reach out with whatever questions you have, whatever you want to start, or wherever you need help or input for. And so this is for Boston, but also through the ACTG. You've seen how many people are connected that actually are in Boston, but you know have leadership roles in the ACTG and other networks as well. So so I think please utilize this, and both these sessions from December and from today were recorded and will be posted on the webpage, on the CIFAR webpage, for people who missed parts or want to review some of that dense information that was provided to help them in their future endeavors. And I think with that, I'm gonna close today's sessions. I thank the speakers again. That was fantastic. Thanks for doing that. And I wish you all a good end of the day. Bye.